This is one of the most searched questions in cancer biology right now, and the answer is more nuanced, more important, and more actionable than almost anything you have read on the internet about it. Because the popular version of this question, does sugar feed cancer, gets simplified into either a yes that terrifies people into eliminating all carbohydrates, or a no that dismisses a genuinely important biological relationship and leaves people completely unprotected. Neither answer is accurate, neither answer is complete, and neither answer gives you the information you actually need to make decisions that could affect whether cancer develops in your body or whether cancer that already exists has the biological environment it needs to grow. So today, we are going to go through the full scientific truth, the mechanisms, the research, the nuance, and the specific actionable conclusions that the evidence actually supports. Stay with me to the end, because the last thing I am going to discuss is the specific type of sugar that the research most consistently links to cancer promotion, not glucose, not fruit sugar, a specific form of sugar that is in almost every processed food on the market, that your body handles in a completely different way from other sugars, and that creates a biological environment specifically favorable to tumor growth through mechanisms that most people have never heard explained. Let me start with the biology that makes this question so important. Every cell in your body uses glucose as its primary fuel. Your brain runs almost exclusively on glucose. Your heart runs primarily on glucose. Your muscles run on glucose. And cancer cells also run on glucose. In fact, cancer cells are extraordinarily dependent on glucose, more dependent than normal cells, in a way that is so fundamental to cancer biology that it has become one of the primary targets of cancer research and one of the primary tools used to detect cancer in the body. This phenomenon is called the Warburg effect, named after the German biochemist Otto Warburg, who first described it in the 1920s. Warburg observed that cancer cells metabolize glucose in a fundamentally different way from normal cells. Normal cells, when oxygen is available, burn glucose through a highly efficient process called oxidative phosphorylation that produces approximately 36 ATP molecules per glucose molecule. ATP is cellular energy currency. Cancer cells, even when oxygen is abundantly available, preferentially use a much less efficient process called aerobic glycolysis that produces only 2 ATP molecules per glucose molecule. This seems counterintuitive. Why would cancer cells choose a less efficient energy production process when a more efficient one is available? The answer is that cancer cells are not just trying to produce energy, they are trying to grow. And aerobic glycolysis, despite being energy inefficient, produces the carbon building blocks, the nucleotides, the amino acids, the lipids, that rapidly dividing cells need to build new cellular material. Cancer cells have hijacked glucose metabolism to prioritize growth over energy efficiency. A study published in Science found that cancer cells can upregulate glucose transporter expression by up to 10 times compared to normal cells. 10 times more glucose transporters on their surface, pulling glucose out of the bloodstream at dramatically higher rates. So yes, cancer cells use enormous amounts of glucose. They are, in a very real biological sense, dependent on it. Now here is where the nuance becomes critical, and where the popular answer gets dangerously incomplete. The question is not whether cancer cells use glucose. They do. The question is whether the amount of glucose available in your bloodstream meaningfully affects cancer cell growth in humans living normal lives. And the answer to that specific question is more complex, because glucose is not optional for your body, it is essential. Your brain will not function without it. Your red blood cells cannot survive without it. Your body will break down muscle protein to produce glucose if dietary supply is inadequate. You cannot simply eliminate glucose from your body, nor would you want to. The relevant question is not whether to eliminate glucose. It is whether chronically elevated blood glucose and the hormonal environment that creates, Specifically, high insulin and high insulin-like growth factor 1 creates conditions that favor cancer development and growth beyond what would exist with optimal blood sugar regulation. And to that question, the research has a much clearer answer. Insulin is the hormone produced by your pancreas in response to rising blood sugar. Its job is to open the door of your cells to let glucose in. But insulin does something else that is critically important to cancer biology. Insulin is a growth factor. It binds to insulin receptors on cells and activates growth and proliferation signaling pathways, including the PI3K-AKT-mTOR pathway, which is one of the most frequently activated oncogenic pathways in human cancer. Many cancer cells have abundant insulin receptors. When insulin is elevated, it is directly stimulating the growth machinery of cancer cells. A comprehensive meta-analysis published in Diabetologia examining data from over 800,000 individuals found that people with type 2 diabetes, which is characterized by chronically elevated insulin and blood sugar, had significantly higher risks of multiple cancers, including liver cancer at 100% increased risk, pancreatic cancer at 82% increased risk, endometrial cancer at 97% increased risk, colorectal cancer at 30% increased risk, and breast cancer at 20% increased risk. These are not marginal associations. These are large, consistent, biologically plausible risk elevations linked to the metabolic state of chronic hyperinsulinemia and hyperglycemia. Insulin-like growth factor 1, IGF-1, is a related growth hormone that is produced in response to insulin signaling and elevated blood sugar. IGF-1 is an even more potent growth stimulator for cancer cells than insulin. It activates the same oncogenic pathways. It inhibits apoptosis, which is the programmed cell death that normally eliminates damaged or mutated cells before they can become tumors. And it promotes angiogenesis, the formation of new blood vessels that tumors need to grow beyond a microscopic size. A study in the International Journal of Cancer found that elevated circulating IGF-1 levels were significantly associated with increased risk of premenopausal breast cancer, prostate cancer, and colorectal cancer. The sugar and cancer connection is therefore not primarily about glucose directly feeding tumor cells in isolation. It is about the hormonal environment that chronic high sugar intake creates. High sugar diets chronically elevate insulin. Chronically elevated insulin elevates IGF-1. Elevated insulin and IGF-1 together create a biological environment that promotes cancer cell proliferation, suppresses cancer cell apoptosis, promotes tumor vascularization, and suppresses immune surveillance of early tumors. This is the mechanism. It is well documented. It is biologically coherent, and it is directly modified by the amount and type of sugar in your diet. Now let me address the specific claim that eliminating all carbohydrates prevents or treats cancer. This is where the nuance becomes most important. The ketogenic diet, which dramatically restricts all carbohydrates including vegetables and fruits, has shown interesting results in some cancer research contexts. The rationale is that by eliminating dietary glucose, you starve cancer cells of their preferred fuel. Some preclinical studies in animal models have shown tumor growth inhibition with ketogenic diets. Some case reports of human cancer patients on ketogenic diets are intriguing, but the clinical evidence for ketogenic diets as cancer treatment in humans is limited, preliminary, and complicated by significant practical challenges. A study published in Clinical Nutrition examining the effects of ketogenic diet on cancer patients found mixed results depending on cancer type, patient condition, and specific protocol used. The more evidence-based conclusion is not that all carbohydrates are dangerous for cancer risk. It is that the quality and glycemic impact of carbohydrates matters enormously. Foods that produce rapid, large spikes in blood glucose and therefore large insulin surges create a more cancer-promoting hormonal environment. Foods that produce slow, modest, stable glucose responses create a less cancer-promoting hormonal environment, even though both contain glucose. The difference between eating white bread and eating lentils is not that one contains glucose and the other doesn't. Both eventually become glucose. The difference is the speed and magnitude of the blood glucose response, and the correspondingly different insulin responses, and the correspondingly different IGF-1 responses, and the correspondingly different cancer biology those hormonal environments create. A study published in the American Journal of Clinical Nutrition specifically examined glycemic index, which measures how rapidly a food raises blood sugar and cancer risk. The researchers found that high glycemic index diets were associated with significantly higher risks of colorectal cancer, endometrial cancer, and pancreatic cancer compared to low glycemic index diets, after controlling for total carbohydrate intake. Meaning the glycemic quality of carbohydrates, not just their quantity, independently affected cancer risk. Eating the same amount of carbohydrate from high glycemic sources versus low glycemic sources produced different cancer risk profiles. This is a critical finding. It means that replacing white rice with brown rice, commercial bread with sourdough, whole grain bread, instant oatmeal with steel-cut oats, commercial cereals with plain whole oats. These substitutions, which maintain total carbohydrate intake but dramatically reduce glycemic impact, have meaningful cancer prevention implications, without eliminating any food group, without extreme dietary restriction, through thoughtful substitution of food quality within the same categories. Now let me address the specific type of sugar that the research most consistently links to cancer promotion. Not glucose from complex carbohydrates, not the natural sugars in fruit which come packaged with fiber, antioxidants, and phytonutrients that modify their biological effects. Fructose. Specifically the industrially produced fructose found in high fructose corn syrup and added sugars in processed foods. Here is why fructose is biologically different and specifically concerning for cancer. Fructose is metabolized primarily by the liver. Unlike glucose, which is taken up by cells throughout the body, fructose is directed to the liver for processing. In the liver, fructose bypasses the primary rate-limiting step of glucose metabolism. It can be metabolized very rapidly without the usual feedback controls that limit glucose metabolism. This rapid, unregulated fructose metabolism in the liver leads to several cancer-promoting outcomes. First, it promotes de novo lipogenesis, the production of new fat from fructose. This leads to liver fat accumulation, non-alcoholic fatty liver disease, and the systemic lipid dysregulation that drives visceral fat accumulation. And visceral fat, as we have discussed, creates a pro-cancer hormonal environment through its production of inflammatory cytokines and estrogen. Second, fructose metabolism produces uric acid as a byproduct. Elevated uric acid promotes inflammation, reduces nitric oxide availability, and creates oxidative stress in tissues. Third, and most recently and most alarmingly, a study published in Nature found that fructose can be used directly as a fuel source by certain cancer cells, and that fructose metabolism in cancer cells specifically supports the synthesis of nucleotides needed for DNA replication during tumor growth. The researchers found that colon cancer cells were particularly capable of utilizing fructose through a specific metabolic pathway that glucose does not activate, suggesting that fructose is not merely a glucose equivalent for cancer cells, but may have specific tumor-promoting metabolic effects beyond those of glucose alone. A study in cell metabolism found that dietary fructose, particularly from added sugars in processed foods, was significantly associated with tumor growth in mouse models of cancer, and that the mechanism involved direct fructose uptake by tumor cells as well as systemic insulin-promoting effects. So what does all of this mean practically? Let me give you the evidence-based conclusions that the full scientific picture supports. Conclusion 1. You do not need to eliminate all carbohydrates or glucose to reduce your cancer risk. Your body requires glucose. Extreme carbohydrate restriction is not supported by evidence as a general cancer prevention strategy for healthy people. Conclusion 2. You should dramatically reduce your intake of added sugars and refined carbohydrates. Not because glucose itself is uniquely dangerous, but because high glycemic foods chronically elevate insulin and IGF-1 in ways that create a pro-cancer biological environment. Conclusion 3. You should specifically minimize high fructose corn syrup and added fructose from processed foods. The emerging research on fructose's specific cancer-promoting metabolic effects makes it the most concerning form of dietary sugar for cancer biology. Conclusion 4. Whole fruits are not the problem. The fiber, antioxidants, and phytonutrients in whole fruit dramatically modify the glucose and fructose impact of fruit. Population studies consistently show that whole fruit consumption is either neutral or protective for cancer risk. Fruit juice, which removes the fiber, is a different story. Conclusion 5. Maintaining stable blood sugar and insulin sensitivity is one of the most important cancer prevention strategies available. Through regular physical activity, adequate sleep, stress management, and a diet based primarily on vegetables, legumes, whole grains, proteins, and healthy fats, you create a hormonal environment that is measurably less favorable to cancer development. The research on sugar and cancer does not support the fear-based conclusion that eating a piece of fruit or a bowl of rice is feeding your cancer, but it strongly supports the conclusion that chronically elevated blood sugar and insulin from a diet high in added sugars and ultra-processed foods creates a biological environment that makes cancer more likely to develop and more likely to grow once present. The difference between these two conclusions is everything. One leads to unnecessary fear and extreme restriction. The other leads to specific, practical, evidence-based choices that meaningfully shift your cancer biology in the right direction. Every single day, starting with your next meal. I want to hear from you. Comment below and tell me, has a doctor or a family member ever told you that sugar feeds cancer? What were you told to do about it? And did the explanation you received match what I just described? Be honest, because the gap between what people are told about sugar and cancer and what the research actually shows is one of the most important gaps in cancer prevention education. And closing that gap starts with conversations like this one. We are building a community of people who understand cancer biology at the level of evidence, not at the level of fear, and who make their food choices from that understanding. If this video helped you, please like and subscribe because we are sharing this kind of evidence-based cancer science every single week. Information that goes beyond the simplified answers, that respects your intelligence, and that gives you the actual biological truth you need to protect your health. Take care of yourself. You absolutely deserve it.